I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. I'm your host, Jesse Lawler, excited to bring you the 55th installment of this podcast dedicated to the betterment of your own brain by any and all means at your disposal. I'm going to be talking this week with a professor from Australia who I suspect will be a repeat guest on Smart Drug Smarts because he's studying so many different things that are of interest to us. Our kickoff topic we're going to be talking this week, it was a wide-ranging conversation, but what we wound up sort of settling on was plant-based cognitive enhancers, and particularly cognitive enhancers that have chronic rather than acute effects, something that you're not necessarily going to feel a quick spike of subjective effects from right when you take something, but the kind of thing that would build up in your system over a long period of time and have ongoing effects that would be measured not in hours, but in months, years, things like that. Also, I've got to offer a blanket apology if I'm not sounding like my normal springy self this episode. And this episode's about a day and a half later published than I wanted it to be. I got a nasty case of food poisoning a day or two ago, which is pretty much my fault. I ate something that had been sitting out a little bit too long, and I should have been more careful. But yeah, just destroyed myself gastrointestinally and am finally starting to feel like a human again. But let this be a lesson to all of you, because boy, if there's one thing that's an anti-nootropic, it is food poisoning. I was running a fever. I was in stomach agony, couldn't hold anything down, couldn't concentrate on anything. And one of the weird, frustrating things about trying to eat healthy, it's like I've been eating really natural foods for probably almost 10 years now. For a while I was vegan and now I'm eating on the paleo diet. But, in all of those cases, I've been really steering clear of heavily processed foods. But the problem with the nice thing about processed foods is that it doesn't tend to rot. And I've probably gotten food poisoning since I've started eating quote-unquote healthily. I'd say probably 4 to 5 times more than I ever got it when I was eating like a regular person. So sometimes you just can't win. If you hang around until the end of the episode, I came across a story that this is not like a try it at home. This is just a weird story. This is a very, very positive story about methamphetamine, or at least positive in kind of the awesome Chuck Norris type of way. Probably completely irresponsible of me to say anything awesome about meth, but I'm going to anyway. The story just was too good not to share. But as usual, let's kick things off with This Week in Neuroscience.
Smart Drug Smarts. This Week in Neuroscience.
So in the U.S. alone, there are an estimated 10 million people that are either blind or at legitimate risk of going blind, or at least legally blind, based on things like macular degeneration and the aging of baby boomers. And it's a really, really big problem. There's a neuroscientist named Sheila Nierenberg at the Weill Medical College at Cornell University that is about a year and a half now from human trials on something which sounds really, really amazing and revolutionary as far as the ability to potentially reverse blindness. So what she's done is she's pretty much cracked the mathematical code of how light signals that hit the photoreceptors in the retina are then transmitted into the optic nerve and then further on into the brain. So what happens a lot of the time as people go blind is it's kind of the front of this chain, the part that's actually capturing light images and turning them into electrical signals. That's where the breakdown happens. Those catcher's mitts for photons cease to be able to catch photons, kind of can't pass the baton of information further up the chain to the actual brain. So while the brain's still functioning normally and even the optic nerve is still functioning normally, it's kind of that front end that's failing. So what Dr. Nierenberg has succeeded in being able to do within the lab is create a way for a digital camera to pass correctly encoded information to the optic nerve that's basically speaking the brain's language, saying, here's the electrical signal analog of what this image out there in the real world looks like, in the same way that the photoreceptor cells would have previously said that. But yeah, very exciting stuff for anyone facing vision loss or who has family members or loved ones that are in that situation. One thing worth noting about this is I'm pretty sure that this treatment would only work for people that were once sighted. It wouldn't work if somebody was born blind, because then their optic nerve, I don't think, would have ever developed the knowledge of how to process incoming signals. So I think, although they didn't specify in this article, that this would only be of use to formerly sighted people. I could be wrong about that, but that is my educated layperson's guess.
Smart Drug Smarts.
So Professor Con Stough is a researcher at the Swinburne University of Technology in Australia. He specializes in psychology, brain science and health, emotional intelligence, theories of intelligence, psychopharmacology and its effects on both cognition and emotion, and pretty much everything that the Smart Drug Smarts listening audience is into. So I was very happy to arrange this interview with him, and as I said, I think that he'll probably be back for more in the future. But he is deep into the research end of research science, and we started off talking about that on the very many studies that they have going on at any one time at Swinburne. This conversation focuses on plant-based cognitive enhancers, but in the future we might be talking about things like his work with typically illegal drugs for medicinal purposes, the Genos Emotional Intelligence Test that Professor Stough and a partner co-developed, and a great range of his other work. So we'll roll back the Wayback Machine here for the interview that was actually a couple days ago. Normally I do these segment recordings almost immediately after the interview, but in this case, food poisoning intervened, as I mentioned. So here we go, jumping back into the interview.
I guess our interests as a research centre, so I'm from the Centre for Human Psychopharmacology, we're interested in pharmacological agents that might improve, possibly even impair cognition. And we use drugs and the pharmacological approach because it tells us a lot about the brain processes and therefore the biological basis of cognition and human intelligence. So we're very busy here doing lots of different drug trials. They are pretty well all randomised controlled trials. We've probably done 50 over the last few years and even more before then. And we probably have at least a dozen clinical trials going on at any one time using what we call both acute and chronic cognitive enhancers and a sort of stack of research around those, including obviously cognitive assessments, but also a lot of brain imaging, a lot of biochemistry and genetics as well. So really very interested in understanding the mechanisms by which we can enhance cognition.
When you're finding your research subjects, how do you go about assessing, I guess, what you consider to be their baseline intelligence to know if you're doing an apples-to-apples comparison?
So most of the trials we do, as I say, randomized controlled trials. So there's a few little tricks in those trials. First of all, they all have placebo. So sometimes there will be a parallel groups design, which means that half of the group will take placebo and the other half will take the active intervention. And we'll compare cognition before and after each of those drugs or interventions across those two groups. So that's the sort of control there. Usually these days we do a lot of computerized cognitive assessments using very validated measures. Sometimes we use paper and pencil, sort of older neuropsychological tests, depending on what we're trying to look at and, the age group and all those sort of things. So in our centre, we've got trials with children, we've got trials in middle age and young people, and we've got trials with older people.
What would be an example of something that you could do with paper and pencil for sort of generalized testing that you couldn't do with a computer nowadays?
Well, that is a good question, and nobody really knows the answer to that. So there are a lot of computerized cognitive batteries now. So we use something called CDR, which has been used in about 1,000 drug trials. We use something called CANTAB progressively, which comes out of the UK. But there's a very big literature on cognition, obviously before the advent of these cognitive batteries. And some of them are derived from patients that have had some damage to the brain. So these are called neuropsychological tests. And they tell us something very specific about certain types of memory or planning or executive functioning. Generally, the computerized batteries these days also try to tap into them as well. And the advantage of computerized batteries is they have very sensitive recordings, particularly of timing. So you can measure reaction time to a millisecond accuracy, whereas paper and pencil tests, obviously you can't do that. You have to, we can measure response times in the order of, seconds, thousands of milliseconds. So there's a lot of advantages using computerized batteries, and there's a lot of disadvantages in using neuropsychological tests. You can only test one person at a time. The timing is much more difficult, and they tend to be pretty complex, and sometimes you'll be picking up sort of qualitative information from them as well. But I would say 90% of all the data we collect now are using computerized cognitive testing. That's not to say that they are the best. We've just written some papers looking at sort of the structure of what we call human intelligence using some pretty famous models like the Cattell-Horn-Carroll model of cognition or intelligence, which is a very comprehensive model of cognition, which a lot of the computerized drug cognitive batteries only measure parts of. And so that's sort of a gap in the literature which we need to look at, I think, for in the future, which we're starting to do.
And that's something that you're probably addressing a lot in your work on emotional intelligence also, right?
Well, yes. So cognitive intelligence is only one type of intelligence. It's started off about 100 years ago with the first IQ tests by Binet in France. And obviously IQ tests have a very long history in psychology, and cognitive testing also parallels about the same sort of timing. So the idea that we might also measure different types of intelligences is very interesting to us. And so emotional intelligence, which is as a construct has only been around for probably 15 years, tries to look at how emotions affect our thoughts, and also how our thoughts might affect the way in which we manage emotions. And obviously, cognition, intelligence, and our emotions are not separate things. They influence each other. So it's a very interesting construct. And the more research has been done on it, I think the more important the role of our emotions are in our everyday success. You know, how we manage our relationships with people. Even how we manage anxiety will be very important in terms of our learning and how we function. So this other construct is very important, but it's really early days in terms of that construct compared to its bigger brother, cognition or intelligence.
Yeah, it seems like as far as outcomes in the real world for how people actually fare out in society, oftentimes emotional intelligence seems like it can trump in many ways pure cognition.
Well, I think it depends on what you're looking at. If you're looking at scholastic performance, then no, cognition is probably more important. But we are finding that emotional intelligence can also add to the predictive variance in scholastic performance as well. If you're looking at how well you do maybe in an exam or what type of job you get, then cognition and IQ have been very predictive of those sorts of areas. But if you're looking at things like quality of your relationships or your well-being, life satisfaction, effective leadership, how well you do once you're into an organisation, so not perhaps getting yourself into an organisation, but maybe how well you do once you're in an organisation. That is, how quickly you get promoted, how quickly you can make effective relationships within that organisation to show leadership and those sorts of things. Then emotional intelligence is incredibly important. And so, one of the things that we're doing, particularly in schools, is trying to develop programs around developing emotional intelligence. I think that's the time to start those sorts of learnings. In terms of psychopharmacology, we're probably quite the same sort of topic, but, very important that I think we think broadly in terms of human intelligence.
So let's circle back a little bit to cognitive enhancers and some of the overall differences between acute versus chronic cognitive enhancers. Can you make some general statements as to what's going on neurophysiologically between those two categories?
Yeah, so I think you have to have a little bit of an understanding of sort of brain processes to sort of understand that distinction. In terms of psychopharmacology, there are a lot of targets in the brain. So cognition is obviously to some extent related to quite a few different pathological and physiological processes. Some of those processes are changes in neurotransmitter activity. Some of those processes are related to receptor activity in those neurotransmitters. So when I talk about neurotransmitters, I talk about things like acetylcholine and dopamine and serotonin that probably people have heard of before. So some of the acute cognitive enhancers try to target changes in these neurotransmitters. And so neurotransmitter activity can occur pretty quickly after ingestion of various what we call acute neurotropics or acute cognitive enhancers. But there are other methods also. So there are some cognitive enhancers that might increase blood flow to the brain. The brain is one of the most greedy organs in the human body for oxygen. Obviously, increasing blood flow means that you can get more oxygen to parts of the brain that are involved in cognition or learning or recall or memory or attention or whatever component you're looking at. Potentially an acute cognitive enhancer would also increase the fuel to the brain as well. So there are examples of acute cognitive enhancers which work typically within relatively small time frames. They will have a half-life of anything between a few minutes to probably a day. Nicotine, for instance, would be an example of an acute cognitive enhancer. It has a half-life of, might be somewhere between 15 and 30 minutes, for instance. And so, after people have a cigarette or a cigar or something like that, you get nicotine into the brain, and you get an increase in nicotinic receptor activity, which is part of the cholinergic system. And you see changes in pretty reliable changes in cognition due to that. We've been also working on chronic cognitive enhancement. So you have to think about different targets in the brain in terms of chronic. So chronic enhancement might mean that we'll enhance the activity of the brain more slowly. So what we might do is we might change the inflammatory response to the brain over a period of time. We might find some agents that might increase the antioxidant status of the brain, or there might be some longer-term genetic signaling changes that occur because of chronic enhancement. Or we might change receptor binding, or we might change the way in which a cell works over a longer period of time. These are the sorts of targets that we're looking at, are often the targets that are associated with cognitive aging. I guess everybody's sort of aware that probably as we get older, there are some cognitive changes that occur. And these occur particularly in memory, but also in processing speed in the brain as well. So what we're trying to do is trying to ameliorate this sort of cognitive change. And these cognitive changes are thought to reflect changes in brain physiology over time.
Why do you think that is? Just out of curiosity, that seems to be a dividing line, that more pharmaceutical ones tend to be the more acute, shorter half-life, whereas it's the natural ones that are on the opposite end of the spectrum.
Well, I think that's part of the whole sort of process in terms of the pharmaceutical industry, is that a lot of these enhancers are not developed specifically for cognition, but are developed for potentially to sell in various diseases and disease states. And sometimes they're sold, for other reasons. The same neurotransmitters in the brain are often found in other parts of the body as well. It's hard to really know, but the pharmaceutical industry will look at very specific targets and develop drugs just for that target. And we've got, a lot to learn about plant-based molecules. A lot of drugs are derived from plants. Something like 60% of all of our drugs are derived from plants. And then they find potentially an active ingredient out of potentially hundreds, and then they'll concentrate that up into a pharmaceutical.
So some of the specific chronic cognitive enhancers that you've studied, you mentioned Gingko biloba as one of those that's fairly well known, and then also Bacopa. The second word in Bacopa I'm never quite sure how to pronounce, so I'll leave that one to you.
Yeah, Bacopa monnieri. It's a very, very old Indian herb. It's probably the oldest herb. I think the writings go back for 3,000 or 4,000 BC, and has historically been used as a cognitive enhancer back in India. So yeah, they're two very prominent chronic cognitive enhancers we've been looking at. There's been a lot of research on Gingko biloba. I'll start off by saying this: if people are interested in plant-based cognitive enhancers, is to be very careful about which ones they buy and the quality that they buy. So if they go down to the pharmacy or somewhere that sells Gingko or Bacopa, they may find that there's 100 different Ginkgos that they can buy, and each extract will be different. Each extract will cost a different amount of money and will show sort of different quality in terms of the extract, where it's grown, the amount of the same active ingredient, whether it has heavy metals or other toxins potentially in it. And often there's only research based on a few of the extracts, and you can't infer the same sort of efficacy from one extract to another extract if it hasn't been subjected to research, because they're quite different. And that's a big difference between the plant world and the pharmaceutical world, because in the pharmaceutical world you have an ingredient and it's the same shape, a molecular shape, whether Pfizer makes it or whether it's a generic brand.
I want to go back to something you said a moment ago, which was heavy metals. Now, I think of heavy metals as not necessarily being something that would be an impurity in plants, but am I mistaken there?
Well, look, they can show up in extracts. So there may be these sorts of toxins in the ground, and therefore they may find their way into the plants and into the extracts. If they're grown in a certain part of the world, which may have more of this, and they're not extracted or carefully extracted out, then it potentially could be a problem.
So what should consumers know, or what can a consumer reasonably be expected to remember when they're putting their shopping list together for knowing what sort of extracts they should be looking for these different things?
Yeah, I think it's really tough because governments don't help them with various laws. Governments in the US and Australia and probably and Canada and so on, not very careful in terms of allowing the markets to be flooded with brands that have not been subjected to research and don't have a lot of quality controls potentially. So I think consumers have to ask questions about the quality of the extracts and what research has been done on them, to be honest. And that's not easy because, extracts can vary considerably in price. So it would be very easy to buy a $10 Ginkgo instead of $30 or $40 Ginkgo. And the question is it going to be effective if you bought the cheaper one? And there's really one or two extracts in the world that have been subjected to very rigorous research. One by a company called Schwabe, extracts called EGB 761, and there's really been a lot of research on that product. And then there's another product that comes out of Switzerland, and one of them is called Gingko San, which is also a very good product, and it's been subjected to quite a lot of research as well. So they're the sort of two main Ginkgo extracts. And I think the evidence for Ginkgo is a little bit mixed as a cognitive enhancer for just healthy, normal people.
Ginkgo is one of these things that's been around as a folk remedy since time immemorial. What was the mythology of Ginkgo before there was the science of Ginkgo?
That's a good question about how these become cognitive enhancers before science. And people historically have experimented with different extracts for different disorders and diseases, and people relatively just stumble upon them.
It's interesting because when you look back into history, there's so many absolutely ludicrous things that people used to do thinking that they were a medicine. Then when you see something that actually has survived into the modern era, it's almost like a miracle. Like, wow, they were actually right back then.
And it's particularly interesting if you go back to 2,000, 3,000 years ago, and people were talking about some of these things as cognitive enhancers, obviously not with the same sort of terminology, but nevertheless in the same way back then. And they had sort of rudimentary ways of extracting the various agents and so on. A lot of the things that we are looking at the moment, such as Ginkgo and Bacopa and some other extracts, things like ginseng, for instance, curcumin, have some sort of traditional use and knowledge as cognitive enhancers, because there are literally so many different plant extracts. It will take us so long to just go through all of them and see which ones are potentially ones that we could use to change cognition. What does help is things like Ginkgo and Bacopa have had a huge amount of in vitro and animal research. So what that does is it tells us about some of the mechanisms potentially on the brain. And then we can map those mechanisms onto the mechanisms by which we understand cognitive enhancement to happen. Bacopa, for instance, there's probably 50 years of very, very solid government research in India. Not so many human trials in India, but a lot of animal and what we call in vitro research, test tube work. And they found that Bacopa has a very strong anti-inflammatory effects, seems to affect beta amyloid, like most plants has strong antioxidant effects, and a range of other processes which potentially map very, very much on cognitive decline and on cognition in general.
And I've heard that one of the interesting things about India as a country is, despite the fact that in many ways it's a fairly polluted environment, that the population has almost a suspiciously low amount of Alzheimer's and senior dementia, that's one problem that they don't have, despite a lot of other public health issues.
People do talk about that. It's possible. It is hard to know, though, because the assessment of dementia in those sorts of countries is difficult. The other thing is that dementia is particularly more likely as we get much older. If a cohort or a country has shorter life expectancies, then you potentially would expect less incidence of dementia. There are more than a dozen replicated studies showing that particularly certain extracts of Bacopa seem to improve cognitive performance in healthy people.
As somebody who's been researching this stuff for years, which of these things has passed the threshold for you where you're convinced and you've worked this into your own diet and the diet of your families and things like that?
Well, certainly the Bacopa that we do research on here, many of us take, and I give to my family and to my friends who use it. They say that they notice some sort of subjective benefits, but in science we don't really accept subjective benefits. But nevertheless, my reading of the literature and our own research suggests suggests that Bacopa potentially could be very useful in terms of protecting the brain from aging and improving cognition.
Curcumin's probably the best tasting cognitive enhancer. That's actually like just a good spice, even if it wasn't doing anything good for your cognition.
The problem with curcumin is that generally it's not very bioavailable. What I mean by that is that it doesn't easily get into the bloodstream in a form that can be helpful. So you have to take a lot of it to get a little bit of it, unless you're using an extract which becomes more bio.
Or, okay, what's the threshold at which something has efficacy goes to something is actually dangerous? For some of these things that have the long-term chronic effects, whether it's Gingko or Bacopa or something like that, it seems like probably what people should be concerned about is more, am I getting my money's worth if I buy this supplement or take the time to find it in the supermarket? Is it going to do something versus nothing? Is that a mistaken opinion? Is there a point at which you could be eating a dangerous amount of Ginkgo, for example, or is that not so much a real threat?
Yeah, no, I think it is a threat. Look, if it's pharmacologically active to improve cognition, then it potentially can affect other parts of the body, of the brain. It can interfere with medications. There are sometimes side effects from plant-based molecules. And there's not a lot of evidence, but people talk about, for instance, Ginkgo, because it thins the blood, much like things like warfarin. So if you take Ginkgo and warfarin together, you're getting a lot of potentially blood thinning, and that could be dangerous. There's not enough research probably on these plant-based herbals and extracts for us to understand the long-term safety effects of them. So we need more studies, but generally when people administer these things for long periods of time, generally they don't find any increases in side effects. So the really long-term Ginkgo studies, one that was conducted by the National Institute of Health, for instance, a few years ago, looking at whether Ginkgo, EGB 761 was the extract, whether that protects people from getting dementia. The efficacy showed that it didn't, but also the evidence showed that there wasn't an increase in side effects versus placebo. So generally, plant-based molecules are fairly safe and very well tolerated, but not always, and sometimes people show side effects.
I've always heard that one of the difficulties just with the way that the pharmaceutical industry works is that it's sometimes hard to find money to do the studies on naturally occurring compounds simply because they're not going to be patentable by a drug company, and therefore nobody wants to pay the money to put them through FDA trials and yada, yada, yada. It sounds like you've managed to find funding for a lot of studies on plant-based compounds. How did that happen?
You make a really good point. It's a big distinction between pharmaceutical research and what we call nutraceutical research. Even though we might be using exactly the same methods, we just use a different molecule. The reason why probably there's so much research being done on Ginkgo is that the German company Schwabi was able to get a patent on its extract, and then they were able to sort of market EGB 761 as something different than most Ginkgo extracts, and nobody else could have the same Ginkgo because it was extracted in a very complex and difficult way.
Is there anything that you'd like to present to the audience or to shine a spotlight on particular research you're doing or links that they might go to see some more of your work that's in progress now?
Sure. I mean, if they go to the Swinburne University website and then look for the Centre for Human Psychopharmacology, or if they search for my name, Stough, Stough, then they should be able to find lots of information on our trials. We're really looking at molecules that affect changes in brain physiology that are associated with aging. And certainly the Bacopa that we've been looking at is particularly interesting to us. And it's an extract called CDR-IO8, which has been used by the Indian government for about 50 years. So we know quite a bit about it. But certainly there are lots of other potential molecules as well. We'll be doing sort of longer trials. I mentioned that we've got a 12-month trial going on with some of these agents at the moment. But, we really need to do 2 years, 3 years, 5 years. Our centre has some very excellent neuroimaging equipment, and we're starting to incorporate things like MRI and MEG into some of these trials at the moment, which is really interesting because it really shows us what these changes are in the brain.
Smart Drug Smarts.
So thank you very much to Professor Stough for taking the time out of his research schedule to let us know what he's been working on and discovering there. A lot of things we touched on, from Ginkgo to Bacopa to curcumin to ginseng. We'll throw up a bunch of links on the website about all these various topics. And yeah, really look forward to having Professor Stough back in the future. By the way, somebody will, I'm sure, write me an email and let me know the correct thing to do here. I'm never exactly sure when to call somebody a doctor and when to call them a professor. Almost all these professors have doctorates, but some of them prefer not to go by doctor, but to use professor as the nomenclature. And I'm not sure if it's just the medical doctors that prefer to use doctor as the title, but anyway, just because you hear me refer to somebody as a professor on this show, don't necessarily assume that there's not a doctorate attached to their title, because most of the time there is. And now, as promised, the ruthless listener retention gimmick.
Smart Drug Smarts. Ruthless listener retention gimmick.
Okay, so this is an apparently true story that happened during the 1980s. In 1986, Namibia was in a war for independence against South Africa, and 2 Namibian guerrilla fighters were on a mission north of the border inside Angola when they realized that they were being followed. They had these trackers called the Kovowits who would track people by their spoor. Spoor is a nice way of saying poop. Anyway, so these 2 Namibian guerrillas realized they were being tracked and broke from whatever their mission was. They're like, we need to get the hell out of Angola. So they split up. They went in different directions. One went north, one went south. The one who went north apparently made an easy getaway. But the Kovowits followed the guy going south. They thought they could get him. They had these things called a Caspier armored personnel carrier, a big, mean-looking, transformer-like truck thing. They were chasing this guy in a completely motorized vehicle. But this guy kept running and hiding and running and hiding, and despite being on foot, he managed to not only stay ahead of, but actually gain ground on the people that were chasing him. During the afternoons, when they could tell that they were fairly close, they would sometimes call in helicopter support. Still couldn't catch the guy. They would occasionally find places where he appeared to have collapsed from exhaustion, but then pulled himself up again. This guy is having no time to sleep, no time to eat. But what they would occasionally find in these places where there was the prints of a human having collapsed were discarded hypodermic needles. Apparently, as this guy was being chased over the course of 5 days with no sleep and no food, he was injecting himself with either Benzedrine or another methamphetamine and jump-starting himself back into physical readiness to outrun motor vehicles. He kept this up for 5 days before making it to a paved road where they lost track of him. They think that he was picked up in a car and was never seen again, successfully escaped. But needless to say, this has nothing to do with nootropics, but it does speak to the rather amazing ability of chemicals to sometimes overclock human biology and make things that seem like they would be completely impossible, like outrunning a motor vehicle for 5 days straight, at least occasionally possible.
Smart Drug Smarts. The podcast's so smart, we have smart in our title. Twice.
You heard it. That is the episode. If you liked what you heard, please recommend this podcast to your friends and or leave us a review on iTunes or write us a friendly email or tweet us to the people that you tweet things to. Any of those things would be great. The show notes for this episode will be online, as usual, at www.smartdrugsmarts.com, including the links to everything that we talked about here. I'll be back at you probably actually in less than a week. It's December 5th as I record this, and I think it'll be before the 12th that we're out again. So even if it's not the same time, it'll be the same podcast and with the same unflagging commitment to helping you fine-tune the performance of your own brain. Have a great week and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.